Welcome to Science for Fun Academy. Today, we'll examine the molecular pharmacology of saffron, Crocus sativus, focusing on its proposed role as a natural monoamine oxidase inhibitor, its pharmacokinetics, and the mechanisms that may explain its antidepressant effects. Before we begin, please subscribe and enable notifications so you're alerted whenever we publish new educational content. You can also support our work through Patreon or by purchasing individual educational resources. Your support helps us remain independent while producing scientifically rigorous content. Let's begin. Monoamine oxidases, MAO, are flavin-dependent enzymes located on the outer mitochondrial membrane that catalyze the oxidative deamination of monoamine neurotransmitters. MAOA preferentially metabolizes serotonin and noradrenaline, whereas MAOB primarily degrades phenethylamine and contributes to dopamine metabolism. Inhibition of these enzymes increases synaptic monoamine availability, forming the basis of MAO inhibitor antidepressants. Among saffron's more than 150 identified phytochemicals, Safranal, crocin, and crocetin are considered the principal neuroactive constituents. Current evidence suggests that Safranal exhibits reversible, predominantly competitive inhibition of MAOA in molecular docking and in vitro enzyme studies. By transiently occupying the enzyme's catalytic site, it reduces monoamine degradation, potentially increasing synaptic serotonin and other monoamines. However, clinically significant MAO inhibition following oral saffron administration has yet to be conclusively demonstrated in humans. Pharmacokinetics are equally important. Crocin is highly water-soluble but exhibits poor intestinal absorption because of its glycosylated structure. During digestion, it is hydrolyzed by intestinal enzymes and the gut microbiota to crocetin, a more lipophilic metabolite that is readily absorbed and appears capable of crossing the blood-brain barrier. Safranal, being inherently lipophilic, is efficiently absorbed and penetrates the central nervous system, making it a plausible contributor to saffron's acute neurochemical effects. Saffron's pharmacology extends well beyond MAO inhibition. Experimental studies demonstrate modulation of serotonin reuptake, attenuation of glutamatergic excitotoxicity, enhancement of GABAergic neurotransmission, suppression of NF-kappa B-mediated neuroinflammation, activation of the antioxidant NRF2 pathway, preservation of mitochondrial function, and increased expression of brain-derived neurotrophic factor, or BDNF. These complementary mechanisms likely act synergistically to promote neuroplasticity and improve mood. Formulation also influences bioavailability. Hydroalcoholic tinctures preferentially extract volatile constituents such as safranal, whereas standardized saffron extracts provide reproducible concentrations of crocins and related carotenoids. Consequently, standardized extracts are generally preferred for both clinical research and therapeutic consistency. Randomized controlled trials and several meta-analyses suggest that standardized saffron extracts improve symptoms of mild to moderate depression, with efficacy approaching that of some selective serotonin reuptake inhibitors in selected populations. However, the available evidence remains limited by relatively small sample sizes, short study durations, and variability in extract standardization. Therefore, saffron should currently be regarded as a promising adjunct rather than a replacement for conventional antidepressant therapy. The key message is that saffron is not simply a traditional herbal medicine. It contains chemically defined bioactive molecules capable of interacting with recognized molecular targets involved in neurotransmission. While reversible MAO inhibition appears to contribute to its activity, Its therapeutic potential almost certainly reflects a multi-target mechanism involving neurotransmitter modulation, neuroplasticity, antioxidant defense, and neuroinflammation. Thank you for watching.